Hi, let's talk about antibodies. This is not the first time that we're talking about antibodies because we have heard about antibodies in the past. In fact, we have said that serology is just an application of immunologic concepts wherein antigen-antibody reactions are involved. And therefore, if we want to understand antigen-antibody reaction, then we need to understand the nature of antibodies. And also, in understanding the nature of antibodies, we would know how they act and how they function as a tool of our immune system. I'm also sure that you have heard about antibodies even when you were still younger, because you would say that antibodies are the ones which kill, and they are the ones which are required with the immune system in order to protect us from diseases. That is why you have vaccines, so that you'll produce antibodies. But let us see how these antibodies look like and how they function. Antibodies are also called immunoglobulins because basically antibodies are proteins and they belong to the globulin component of the protein. Remember that proteins basically have two important fractions. Your total protein is made up of your albumin plus the globulin. Albumin composing the majority of it, while globulin is further divided into alpha, beta, and gamma globulins. The gamma globulins, it is in this portion where you see your antibodies. Antibodies may be either membrane-bound or free antibodies. What do we mean by this? We recall that B cells have antibodies as their receptors. Thus, when antibodies are still attached to B cells, then these are called as membrane-bound antibodies. We also recall that when B cells are activated, they become plasma cells, and plasma cells produce antibodies into the plasma. This is the one which we call as free antibodies. In this diagram, you will see a normal electrophoretrogram, and you'll see here that albumin here has the highest concentration when we try to do an electrophoresis of the serum. And these are your globulin components, the alpha globulin, the beta globulin, and the gamma globulins. It is in this portion where you will see the immunoglobulins or the antibodies. So again, basically, antibodies are actually protein in nature. In certain instances or in certain disease states, sometimes there are so many antibodies present in the system that you see a monoclonal peak here. There is a high concentration of antibodies that you will see in this portion. This is the structure of an antibody. Okay, you have the constant region here, as you will note from this dark blue. Oh, before we go into that constant region, let's look at the chains. This blue chain is your heavy chain, and this pink and red is your light chain. So there are two chains forming the antibody, the heavy chain and the light chain. And each heavy chain and light chain contains a constant region here. For the heavy chain, this is the constant region, the dark blue colored part of this illustration, and a variable region here on top, the light blue colored, for the heavy chain. For the light chain, that is the light pink in color, while the constant region is the red portion in this diagram. What then is the importance of these regions? First, you will notice that the variable region is located in this area of the antibody, which means that this is the part of the antibody which may vary or which may change. Why does it need to vary? Or why does this area need to change? Now, this is the part of the antibody which actually binds to the antigen. Therefore, this is the part that should change in order to fit into the antigen. Thus, this area is known as the variable region. This is the variable region of the heavy chain. This is the constant region of the heavy chain. This is the variable region of the light chain, and this is the constant region of the light chain. These constant regions, as the name implies, are constant, therefore they do not change. Only the variable region of the antibody changes because it is the one which is made to fit into the antigen. But this is just a diagram or an illustration. This one here is a more accurate chemical representation of the antibody. You will see here again, this one is the heavy chain, this one is the light chain. Notice that these two chains are connected by a disulfide bond. That is why they do not disintegrate. This is also the light chain, and this is the heavy chain. And here at the top you will see the variable region, which is the antigen binding site. And these, which are colored darker, are the constant regions. Here in this illustration, you will see the light chain. Okay, this is the variable region of the light chain, and you'll notice that these are the loops that bind the antigen. And these are the loops that change in configuration in order to fit the antigen. This is the constant region of the light chain, which means that this does not change because this is constant. Now, when the scientists were still first studying the antibody, they actually have to divide this into different parts in order to be able to figure out if this part is really binding the antigen and this part is not binding the antigen. Two enzymes which were used in these studies are papain and pepsin. What's the importance of these enzymes? When you use papain to break down an antibody, it acts on the hinge region of the antibody. This part here is the hinge region. What is the function of the hinge region? What is a hinge? You would remember that a hinge is usually that part which you place on a door so that you can open a door. Okay? Therefore, the hinge region is also the part of the antibody which enables this part to move down or up in order to fit to the antigen, to the location of the antigen. Okay? So if you use papain, this is the site of action. If you use pepsin, this is the site of action. Now, what is its essence? What is the significance? When you use papain to break down the antibody, you get these products. Since it acts on the hinge region, you get two of this and one of this. Now, it was found out that these two fragments are able to bind the antigen. Well, normally because this is the part which binds the antigen. These two fragments are called your Fab, or your antigen binding fragment. Well, this part at the bottom was observed to crystallize in solution. This does not bind an antigen. This is now called the Fc region, or the crystallizable fragment. These are the products which we get when we use papain to digest the antibody. We get two antigen binding fragments, it's 1 and 2, and we get one crystallizable fragment, or 1 Fc. Now, the other enzyme is pepsin. If you use pepsin to degrade an antibody, it acts below the hinge region so that you will have these products. You will notice that in this fragmentation, the two antigen binding sites are still connected to each other so that you get a big fragment with two antigen binding sites. Thus, this is how we denote here. If you'll notice FAB2, this is how we denote this fragment, because there are two antigen binding sites in just one fragment, while the other one is not actually a complete crystallizable fragment because they are simply made up of low molecular weight peptides. So, in order for you not to forget the action of papain and pepsin on the antibody, remember that if you just alphabetize the two, papain will appear first before pepsin, which means that papain would act higher than pepsin. So papain acts on the hinge region, giving you 2Fab plus Fc, while pepsin will give you Fab2 plus an Fc prime. So that's it about the structure of the antibodies.